Hello, it is now time to start. Good morning, everybody. Thank you to all of you for attending so early the symposium organized by NAOS on the fundamental discovery, B1, the genome, the proteome. You will discover during this symposium a reveal after years of research, the importance of the proteome that represents all the proteins expressed by our body. And you will see how important is the proteome and how it plays a fundamental role, especially in aging, and how it is necessary to protect this proteome. And to present you this discovery, I have the pleasure to introduce first Professor Miroslav Radman. Professor Radman is one of the leading molecular geneticists. When he was researcher at Harvard, he discovered the DNA self-repair system. Professor Radman was director of research at CNRS, at INSERM. He was professor of cell biology at the René Descartes University in Paris, and is the founder of the Mediterranean Institute of Life Sciences. So thank you very much, Professor Radman, for being here today. It's a real honor for us to have you presenting a proteome-centric view of aging and age-related diseases.
Good early morning to everybody. I'm pleased to be here on an event sponsored by NAOS, because the scientific story that I'm going to try to summarize 15 years of research is linked initially with a friendship with Mr. Jean-Noël Torel, the founder of NAOS, and then later with the whole team, and in the last 10 years have been really a cooperation alliance, common game in doing the research that corresponds to this bombastic title, you know, mechanism of aging and age-related diseases. What I'm trying to do this morning will be just to familiarize you with the key new things, to tell you something that no other laboratory, institute, or scientist will tell you these days about aging. And the surprising news is that the conclusion is that fundamentally you will see aging and even hundreds of age-related diseases are not complicated. That this is the main message that I'll tell now in advance. What is complicated are the consequences of disease and aging, but the process of disease and aging, its cause, you will see is very simple. And if it's true, then it should be simple for us to continue to work and extend the collaborations. It should be then relatively simple, strategically simple, to prevent all of age-related diseases together and maybe slow down the aging process itself. And we are not talking anymore only about the skin, which is very important. I said the cell itself is defined by its skin, which is the membrane. But it is about the whole body, because when we age, the whole body ages. Nothing stays young while other things are aging. So I'll try to pass the message in its simplicity, in its essence, and if in these 15 minutes I succeed, I'll be very happy. And I guess in the pause we can then have short discussions. Okay, so we'll start now with the title and go to the theme, the fundamental mechanisms of aging and age-related diseases. These are the questions. What does time do to cells to make them age and to cause morbidity and mortality? what determines the species-specific longevity, that the worm Caenorhabditis elegans lives 18 days, and we can live 100 years. And also what determines the predisposition to different age-related diseases? What makes us different in face of time? Our destinies are largely predestined, and what is the molecular basis, what is the physical basis of our health-related destiny? All right, so let me shift to this. Don't even, try to look. It's unnecessary to look at the detail. What is necessary is that you see this pale. I don't know why it was made pale. This pale curve here is the curve of today's in France mortality rate per age per year, the whole population, and then you see how many are dead in 1 year after birth, 2 years, and so on. And you will see that the lowest mortality in our lives is already at age of 10. At age of 10, here I'm trying to indicate, let me see, age of 10, the mortality is, this is preparing the organism for reproductive activity. So the quality of human body is the highest at 10. And then it goes exponentially with fifth power, sorry, this won't be, with fifth power, let me try this. No, this one. Well, it's not punctual, but you can see now better this arrow. With fifth power of age, this is the most relevant view on human mortality and morbidity in the case of France here. And fifth power, it means that as we double our age, say pass from 30 years to 60, the risk of dying and having one of these diseases will be multiplied by 32. If we smoke double the number of cigarettes smoked, the risk will double only. But with time it goes with fifth power. Well, the point of wanting to show you this is the following. This is the mortality curve. It increases exponentially. And these spaghettis here are individual diseases, about 100 malignant diseases and about 75 degenerative diseases. And the point of showing it, you see how these terribly, incredibly different diseases, cancer kills by excess of cells and Alzheimer kills by deficit of cells, and the mechanisms are totally different. The effects are totally different, and yet the slope is fifth power of age, as if all of age-related diseases, which is 90% of mortality in France, have all the, sorry, this is something is not in my control. But anyway, as if these amazingly different diseases have all a common clock. And this is important. A common clock, they increase with the same slope. And that means that having common biological clock, that the simplest way is that they have a common cause, common origin. Imagine that all of these different diseases have a common cause, and then explore what is that common cause. I'll tell it in advance. You will see it's not DNA damage, it's not the number of mutations during life. It is the damage to functional molecules of life, damage to proteins. And the point of showing this is that if there's a common cause to aging and age-related diseases, and that is simply corrosion of biological material, that we are sort of fundamentally aging like our cars. Only car is corrosion of metal, and here is corrosion of biological molecules, right? If there's a common cause, then it should be simpler to mitigate, to prevent and cure all of age-related diseases, all 90% causes of death together, than to pick any single disease and say, I want to prevent this disease, right? If the common cause would involve necessarily the whole spectrum. And that's the reason why I came here sort of with this message of optimism, that we may be maybe in 4 or 5 years, we may be able to prevent at least all of age-related diseases. And this is not a stupid dream, because centenarians, people, four persons in 10,000, live exactly that. They're not, through their lives, they don't suffer any of these degenerative diseases, and therefore live 100 years. In a way, the vision would be, the project would be to make us all healthy centenarians, that everybody will live 100 years, right? And why is this dancing the dances that I'm not controlling? Okay, right. So maybe just this, yeah. So these 3 circles here, just very quickly, is analysis by top experts in a review, analysis of what is common to studies of aging in bacteria, yeast, worm, Drosophila, fly, and humans and human diseases called progeria, where the aging is extremely accelerated, and the children of age of 15 look like 90 years old people. And they concluded that the common thing is, here, ROS and tissue homeostasis. We won't talk about tissue homeostasis this time, although in our alliance, NAOS, Medials Institute in Split, we do have a productive research into what is tissue homeostasis, which is sort of, like biological music, harmony. We won't talk about that. We talk today about ROS. ROS is the only thing that is common. So what is ROS? ROS is, you see, produced by metabolism here. You know about this in mitochondria, that during the oxidative phosphorylation, there's release of electrons. And there's this species here, OH, hydroxyl radical, is by far the most corrosive radical of all known radicals. And you can see that by irradiating the living cells, one can produce, through radiolysis of water and so on, free radicals of the same kind as in normal oxidative metabolism. That's the origin of free radicals. Now, when looking in extremely radiation-resistant or long-lived small animals and even bacteria, it comes out that the reason for their extreme resistance, when I say extreme, means surviving 1,000 times higher dose than normal bacterial species or cells, is that we found that the common thing and the cause of their robustness, extreme resistance in like truly death-resistant organisms is the protection of proteins, of ensemble of proteins, which is called proteome, from this corrosion by the free radicals that you see here. And that's sufficient. The story went on, and we had to reorder the basic picture of molecular biology, which is DNA, RNA, proteins. It's called the central dogma of molecular biology. And now this is to show that in the middle of life activities are proteins. We have about 21,000 protein species, which then carry on all life functions, including, repairing, replicating, copying, expressing genes here, and then genes are necessary to, let me show you. Genes are necessary as information to make proteins, and this goes through RNA. Genes are copied first in the sequence of RNA, and the sequence of messenger RNA makes in the ribosomes makes the proteins, and proteins are functions. So we have here the information, which is genes. We have here function, which is proteins, and we have the link between information and function, which is RNA. So it turns out that the activity of proteins is key to life, because there are cells, our red blood cells, erythrocytes, which have no DNA, and yet they function for 2 to 4 months, and very, very much function in playing with the oxygen, which is close to the oxygen free radicals. And so this is a redesign of the importance of these macromolecules in life, that it isn't anymore DNA up here like the boss, the king, the boss of life, and then that is the RNA, and then proteins as some kind of working class molecules. Proteins carry on all life functions, and the genes are important because there's the information, the design to make proteins, to refresh the proteins. Okay, now let me summarize to what we have learned in essence, the basic things. Here's one. This blue circle is the amount of free radicals. It increases with age. It's immediately produced by radiation, if you wish. And what we have done conceptually is to bring together, it turns out that it's brought together this oxidative stress biology and chemistry, and the folding of proteins. The protein is a linear sequence of amino acids, as you know, and there would be one species of proteins if it were not structurally folded and specific. The information is in the amino acid sequence, but that sequence allows interactions and makes some kind of knots and so on, and then that three-dimensional structure determines the specificity, the activity of the protein. So when proteins are, this is now summarizing only, no, I won't give you the arguments that are largely published. What we discovered is that the proteins have evolved during billions of years of evolution with living with energy, with free radicals produced, and that the native perfect fold structure of proteins is practically inox. The evolution did its work. It retained those mutant proteins which were most resistant to oxidation. Others, would be burning out immediately, and you will have to make new protein all the time. The life would be not very efficient without this structure being such as it is. You can enjoy looking at structures of thousands of proteins that are determined and enjoy how different they are. But they have in common that once folded, they are practically inox, not totally, because even the structure of proteins is breathing like this. And when it breathes, the free radicals can damage the protein and lock it in this non-functional state. Now, this is the way many of these, many of these experimental conditions can be found where you have a lot of free radical, normal proteins. You can have normal amount of free radicals, but destabilize proteins by deficit of chaperones, of which chaperones are proteins which are doctors of other proteins, and fix their structure when they are misfolded, pump the energy, and make it to fold correctly. So the misfolded proteins are punished by oxidation for their imperfect structure. And this imperfect structure, by the way, comes also from fixed mutations. And when a single protein species, because of a predisposition, because of a weak link that is inherited, makes the protein sensitive to oxidation, it predisposes to particular disease. And you can all read this in my two open biology papers in 2019. It is described and pictured in relative detail. Okay, so let me run through this essential of aging and diseases. Come on. Okay. Have a look here. This is something that I think you are familiar all. It's just take the picture of summarizing it. Is that the more misfolded, imperfect the proteins are, and the more of free radicals are, the more will be a proteome disaster. And the reasons for aging, for disease, and just have this picture in mind as a summary. As expected, cells that are immortal, they are germline cells, embryonal stem cells. They are by far the lowest in amount of oxidation, of carbonylation, oxidative damage. You can see here that is close to this Deinococcus bacterium, which is indestructible bacterium, and it fits. Immortal cells are immortal because their proteins are well protected, and this is our germline. As soon as the cell differentiate, they're stem cells as well, as soon as they differentiate to do the work, they go in oxidative phosphorylation, and their life is counted. We have to die. So I think that I was too slow, and I will run only through to get to the consequences, final consequences of this protein damage. And here it is. I'm a little slow because I don't awake well early in the morning. And that is here that you have to meet misfolded protein, imperfect proteins, and there are so many reasons when they are imperfect. as soon, like in cancer, there's a speeding up of protein synthesis. And whenever you speed up something, the error rate goes up. Americans say, rush makes trash. But so if you want to be perfect, you have to slow down. You will be inefficient. And so the cells find this sweet point where the efficiency, the speed of producing synthesis and function, and their quality, it has to be balanced, right? And here we have it. We meet misfolded proteins and ROS, and have oxidized proteins. This in the middle there is the cause of all evil that we meet in our lives. This is the cause of diseases. Sorry, I won't be showing you this. Damaged protein is either misfunctional, it doesn't function well, or non-functional, or even worse, it becomes toxic. It makes structures like this. These are like artificial pores. They become sticky like Velcro, and then make the circle, and these are hydrophobic pores that go in the membrane, and this is what kills our cells. This is a general mechanism of cell death, is making holes in the membrane, all right? And here are the consequences. These abnormal proteins are subject to degradation. The proteasome takes care of them. There's a cleaning system, which is also made of proteins, not of diamonds, which oxidize. And with age, the cleaning is less and less efficient, and that causes the level of protein damage to increase exponentially with age, and cause both healthy aging in centenarians. Centenarians, don't look like 20 years old. They age, but they don't have diseases. Okay? And the project here, our project here is to find molecules, and we found them already, in extremely resistant organisms, from bacteria to little animals, that are chaperones. They are simple chemical chaperones that sit on proteins, and they are antioxidants. So they protect specifically the proteins, and they are found in bacteria that are death resistant. So we hope to be able to transfer, you have to be naive to do something always, to transfer this death resistance, to transfer this death resistance from such extremophile or extremely resistant organisms to ourselves, to be egotistic, to take it with a drink, with a pill or something. So, okay, with this optimistic vision, I'll stop boring you, and thank you for your patience this early in the morning.
Thank you, Professor Radman, and we understand no better with your talk the really role of the proteome as a guarantor of cell longevity. But what if the proteome is altered? And I have the pleasure to introduce Isabelle Benoit, member of the NAOS Scientific Committee, for a talk about proteome alteration as a root cause of skin aging.
So good morning, everybody, and many thanks for being here with us today. While this is operating, I would just like to quote what Professor Radman has said, that our stem cells are immortal because their proteome is protected. And this is a good transition with what we are going to elaborate further upon this morning. Professor Radman, thanks for this very inspiring talk with this new scientific paradigm that, I will have a problem with that because I cannot stand still. With this new scientific era, that oxidative damage to the proteome is the root cause of aging. What is true at the level of the body is even more so at the level of the skin, being our skin our largest organ and the most exposed to environmental stress. So this morning, I would like to show that if oxidative damage to the proteome is the root cause of skin aging, could protection of the proteome of the skin be a breakthrough, a step forward for age management strategy and for skin care strategy? I would like to tell a little bit the story about the discovery by Professor Radman, because it's still moving for me to know that the ground of this work dates back from the early days of life on Earth. Indeed, the inspiration of Professor Radman came from the study of longevity heroes. He wanted to unravel the secrets of longevity. Logically, he studied longevity heroes, and more particularly, extremophile organisms, meaning organisms able to sustain, to live and even to thrive in an environment that should kill any form of life. These small organisms, usually bacteria, can sustain the high radiation level, oxidative stress, extreme temperature changes, desiccation. They are quasi-immortal. And they are considered as example of evolutionary perfection because they manage, through successive adaptation throughout their evolution, they manage to change a hostile environment into a suitable one. One bacteria attracted more particularly the attention of Professor Radman is called Deinococcus radiodurans. You know the story of radiodurans. It can survive 3 years in outer space, for instance. It's an experiment conducted in ISS in 2020. These bacteria can survive doses of radiation that kills human beings. So the bacteria is like that, and you see us, and it can sustain higher doses of radiation. Being a geneticist, Professor Radman thought that the reason of such a robustness was at the level of the genome. So he tried to find explanations what made the DNA of radiodurans particularly resistant. And he discovered that the DNA is not more resistant. When it is irradiated, the DNA gets damaged, destroyed even. But it doesn't kill the bacteria because the DNA repairing proteins are efficient, are robust, are performing, and can repair the damage. Hence, this paradigm that longevity is not linked to the genetic codes, and we should find reasons of longevity at the level of proteome protection. The key for long life in good health is protecting the proteome. This is where we start from for our NAOS Aging Science R&D process, and I would like to guide you through that with first a reminder about what is the proteome. Remember that the proteome is to the proteins what the genome is to the genes, which means that the proteome comprises all the proteins in your cell, all the proteins in your body. Proteome comes from the Greek protos, which means first and essential. First, because in any cell and any organism, after water, proteins are the second most abundant element. Today we focus on the skin. Do you know that 70% of the whole body proteins are expressed in the skin? In the skin, you have roughly 70% of water, and you have between 15 and 28% of proteins. It's a lot. And it's a lot because in the skin, as in all cells, proteins play an essential role. They can be either structural or functional. The structural proteins of the skin are very well known. They are the target of our industry for decades, and it's true that they should be the target. Collagen, elastin, keratin, that constitute the scaffold of the skin, the architecture of the skin. But you also have smaller molecules with a functional role. All the life effector of the skin are proteins. You know them. Growth factors, enzymes, cytokines, interleukins, aquaporins, connexins, everything is protein. If our skin is a living world, it is thanks to its proteome, thanks to its proteins. So if proteins provide all the function of life in the skin, it's logical to understand that any damage to the proteome will impair the life of the skin. And indeed, oxidative damage to the proteome is the root of skin aging. Protecting the proteome is hence the key for longevity. We call that a new scientific paradigm. Okay, we don't talk about a new theory of aging. Indeed, for us, it is much more than just a new theory of aging. It is what we call a meta-theory of aging, the mother of all theories of aging. You know all of them. They have been described in 2013 by Otin that describe the biological geriatric assessment concept. In this paper, he lists all the theories of aging. You have heard all of them. These are the buzzwords of the industry for decades now. Telomere attrition, chronic inflammation, mitochondrial dysfunction, genomic instability, and so on. What we posit today is that all these theories can be related to proteome protection. Proteome protection encompasses all the previous theories of aging. I will give you 3 examples. Why do we believe that it makes more sense to protect the proteome than to protect the genome? Well, first, because we learn from nature, and what nature tells us with the example of these extremophile organisms is that when the genome is destroyed or damaged, what repairs it? It is the proteome. So for a long-lasting view, it's better to save the doctor than to save only one single patient. There's another reason which lies a little bit on common sense. As you know, genome comprises all the genes, so it represents everything that could happen. But not all of genes are expressed, and actually many of them remain silent. It's only when a gene is translated into a protein that something happens. So to summarize, we could say that the gene, the genome, is a virtuality when the proteome is a reality. As industrials, for the sake of efficiency, we'd rather focus on the reality. Also, as industrials, rather now on safety and I would say maybe ethical viewpoint, we believe it is safer, less dizzying to target the proteome rather than the genome. We want to remain on the safe side. This is the first overlap between genome and proteome. Second overlap with proteome protection and the telomeres attrition theory of aging. You know this theory that you can correlate the age of the cell with the length of its telomeres. Telomeres are at the endings, at the end of chromosome, in charge of protecting the DNA. Replication after replication, they shorten, which fragilize the DNA. So you want to prevent the attrition of the telomeres. In the cells, this is the role of telomerase. So to prevent the attrition of telomeres, you must protect telomerase. Telomerase is a protein. Unfortunately, it's one of the less abundant in the organism of humans, and it is also the one more sensitive to damage. So proteome protection, if you protect the telomerase, you will prevent the attrition of telomeres. Okay? We also have a link with proteome protection and inflammaging, low-grade inflammation. So this one is a little bit robust for so early in the morning, so I need to have your attention. Okay? So chronic inflammation is initiated by inflammasomes. Inflammasomes are protein complexes inside immune cells. They are in charge of defense against the pathogens. When a pathogen arrives, inflammasomes initiate downstream inflammatory cascade. So pathogens, inflammasome, release of cytokines, interleukins that clear the threat. So this inflammation is beneficial in case of pathogen aggression. But aberrant release of cytokines and interleukins, as you know, induces a low-grade chronic inflammation called inflammaging that accelerates aging. So you want to stop this aberrant release of pro-inflammatory mediators. And there is one effector of this stopping of inflammation, which is called sirtuin. Sirtuin inhibits inflammation, low-grade inflammation, and this sirtuin is also a protein. So if you protect the sirtuin, you will prevent low-grade inflammation, you will prevent inflammaging. And you can unroll this story for all the 9 biologic geriatric assessment concepts, for all the theories of aging. You can make the link with proteome protection. Hence, our conviction that today we are not just presenting a new theory of aging, we really open a truly new scientific era, the era of the proteome. So now that we are convinced, I hope you are convinced that we should protect our proteins, what should they be protected from? The main damage to the proteome is called carbonylation. Carbonylation is a specific kind, it's a special kind of oxidation. So like all oxidations, it is mediated by free radicals, but it targets specifically proteins. It's an oxidation specific to proteins, and it's particularly damaging to the protein because it blocks it in a structure under which it is no longer performing, very difficult to eliminate, and impossible to be repaired. So as you know, proteins, in order to be performing, must be under a specific 3D conformation. In this 3D conformation, the sensitive part of proteins, amino acids, hydrophobic amino acids, are inside the core, protected by the core. When the protein is damaged, misfolded, or unfolded, it opens up and exposes the sensitive amino acids. And these are the target of the free radicals for carbonylation. And once the amino acids are carbonylated, the open protein is blocked. It's a kind of lock. So it cannot be refolded again, and it cannot be eliminated because these carbonylated proteins tend to aggregate. And these aggregates are too large to be eliminated by cell natural detoxifying system. You can correlate the age of the cell with the content in protein aggregates. They are at the same time the marker of aging and the accelerator of aging because they impair the natural detoxifying system. At the level of the skin, because skin is our interest this morning, carbonylation targets all skin layers. At the level of the stratum corneum, carbonylation induces dehydration. Living layer of the epidermis, carbonylation induces loss of inverness, loss of radiance. Dermis level, carbonylation induces wrinkles, loss of firmness, and loss in density. So what must be taken as a message from this slide is if we manage to prevent protein carbonylation, we act ahead of all the age signs. So one single comprehensive approach allows to have an effect on all age signs of the skin. How could we do that? We understand that we must protect on one side the protein, and on the other side, we must neutralize the free radicals that induce the carbonylation. The most efficient approach would be to have this two-pronged mechanism of action, first at the level of the protein, second at the level of the free radicals. At the level of the protein, we just learned from nature, you have molecules in the wild which are programmed to protect the protein. They are called chaperones. They are expressed in basal conditions with a role to assist the proper folding of the protein under this conformation I was talking about. But in case of stress, these chaperones are overexpressed with a role either to refold the protein or to cover it, to hide the sensitive amino acids, to reduce the sensitivity to the carbonylation. So the role of a chaperone is rather like a physical shield. It's a physical hiding of the sensitive part of the amino acids. And then you have the antioxidants that you all know in the cosmetic industry. You know that antioxidants can scavenge the free radicals and reduce the inducers of the carbonylation. So the ideal molecule would be to have both this physical role and biological role. Antioxidant chaperone would be a major breakthrough in age management strategies that could supersede traditional antioxidants that only have this antioxidant effect. The good news is that NAOS aging science found naturally occurring chaperone and antioxidant, where, no surprise, in an extremophile organism. So not the same one as Professor Radman, no Deinococcus rhabdodurans, but another one called Arthrobacter agilis. The nickname is snow bacteria because it was first isolated from snowflakes. It was snowing one evening in Paris, and a researcher collected snowflakes and said, okay, maybe some bacteria survived in these snowflakes. And if they survive, it means that they can protect themselves from high level of radiation, from extreme cold. So let's check what is alive. And what was alive was Arthrobacter agilis. And it's alive because it has learned over the centuries to synthesize the best protectant of its proteome. In Arthrobacter agilis, you find bacterioruberins. Bacterio because it comes from a bacteria, and ruberin because of the striking red color that you see here, which is the true color of the molecule. So these bacterium ruberins are at the same time chaperone-like and antioxidant. So they are the best protectant for the proteome of the bacteria. And a specificity of Arthrobacter agilis is that under this bacteria, bacterioruberins come under 6 different forms. And these different forms of bacterioruberins allow protection on a very wide array of proteins. Thanks to the 6 different grades of bacterioruberins, you can protect most proteins of your skin. It's a little bit as if this bacteria has benefited from 3.8 billion years of evolution to synthesize the ultimate proteome protective system. And this is what we extract at NAOS Aging Science. We extract these 6 bacterioruberins, and I will guide you through some of the few of the tests that we have conducted. This is to demonstrate the chaperone activity. So we take here an enzyme. Sorry. Toujours pas. We take here an enzyme, which is alkaline phosphatase, but it hasn't got any importance. You see that at room temperature, we have good level of this enzyme. If you heat the enzyme, you don't oxidize, you heat the enzyme, you unfold the enzyme. The enzyme is a protein, and it's very sensitive to heat. And you lose the activity. If you treat with Arthrobacter agilis patent, you maintain 80% of protection. And see here, compared with the other antioxidants, only Arthrobacter agilis have this efficacy because it's chaperone activity. And antioxidants do not have a chaperone activity. Okay? Only our extract was able to stabilize the 3D structure of the protein. Then we checked also, this time for antioxidant power, because we have seen that it is the only one to have a chaperone activity, chaperone-like activity. How does it perform in terms of antioxidant power? Well, you see here all the big names of the antioxidant that you know. You have vitamin C, Trolox, which is vitamin E, ascorbic acid, glutathione. You see here that Arthrobacter agilis is a very powerful antioxidant. Chaperone-like antioxidant, logically, it protects the proteins of your skin. We have here made a demonstration on protection of a structural protein from the matrix. So 2 learnings from this test you see here. It was an ex vivo test. Control, we stress the skin explant with UVA and pollution. What you have to monitor is the influence on the purple color, which is the marking of elastin. So you see that the stress induces a loss of elastin density. The control, the placebo, has no effect, but Arthrobacter agilis has an effect to protect the elastin of the skin better than tocopherol, which is vitamin E. Two learnings from this test. First, you have a protection of a structural protein, very important for the industry, elastin. Second, demonstration that it penetrates, okay, because this is ex vivo studies. So we demonstrated that if you apply on top of the skin explant, you penetrate enough to protect the elastin. We also checked the effect on another protein of the skin, this one a functional one, called endonuclease. Alors, I don't know with the accent, but it's T4 endonuclease. It's involved in the DNA repair. If you irradiate this endonuclease here, you lose the activity. Most of the enzymes, most of the proteins are sensitive to oxidative stress, unless you protect them with Arthrobacter agilis patent. So these bacterioluberins maintain the ability of the cell to repair the DNA upon UV irradiation. And this really completes the circle with a nice story of Professor Radman. What do our patents protect the cells from? Carbonylation, but what is the source of carbonylation? We checked various carbonylation inducers, UVA, blue light, and particles. So this is an in vitro test. You have here the cell culture. If you stress them, you induce carbonylation, revealed in red here in terms of fluorescence. So we see that UVA, blue light, and particles, all of them induce carbonylation. But if you protect the cells with Arthrobacter agilis patent, you prevent this carbonylation coming from various sources, UV, pollution, particles. So this is only a snapshot of many of the tests that we have conducted on our raw material. I hope that it allows you to be convinced that this really constitutes a breakthrough, a step forward in age management strategies. In terms of what, we are convinced that protecting the proteome is the new dogma that everybody should really focus on. Why? Because proteome protection is not just a new theory of aging, it is the theory of aging, a meta-theory of aging that encompasses most previous ones. And this proteome protection should be effected by specific molecules, chaperone-like and antioxidant. Being chaperone and antioxidant, they supersede traditional antioxidant approaches. And the good news for all of us, because we are women, most of us, is that this solution is within hand-reached because NAOS aging science has managed to formulate these chaperone-like antioxidant molecule from cyanobacteria in a serum. It will be an Institut Sedam serum, and it will be released soon with the proposal to allow your skin to age twice slower.
Thank you. Thank you, Isabelle. So inspired by the work of Professor Radman, the NAOS research team effectively discovered the way to protect the proteome from damage in order to maintain skin usefulness.
Okay.
To continue on the proteome, yes, I have the honor to introduce our third speaker, Professor Marco Rocha. Marco Rocha is a volunteer professor and researcher at the Sao Paulo University, and he is the director of Marco Rocha Dermatologia. He's also a member of the American Academy of Dermatology and of the Brazilian Society of Dermatology. Okay, and the talk of Professor Rocha will be on the protection on the proteome, a new therapeutic way for healthy aging. Thank you.
Good morning for everyone. Thank you very much, NAOS Foundation. It's a pleasure to me to be here today. And I would like to start with a question. Why not two men? Why just for women? So it's not fair for me because I'm a man and would like to start. Okay? So
thank you, Marco.
Okay, okay, let's do it. Okay, I am a dermatologist. I'm trying to understand more about this question involved proteome. Because in dermatology, we have a notion with skin diseases and aging process. And we're trying to surf in a wave called genes. But this wave is so difficult to surf that we didn't had a great success. So we moved to another wave called proteins. And I hope to have more success using this wave to fight not just the aging process, but even to fight or resolve even better than skin diseases. So, of course, the background is our body has 16% of proteins, but this wave is not so simple because protein is not just a simple molecule, it's a three-dimensional molecule. We need to fold and refold this molecule to get the correct function, and there's a lot of proteins. We have almost 25,000 genes related to more than 1 million proteins. So 1 million proteins in the body, it's not so easy. We need to have the proteome's quality control. And to do that, we have a machine. We have a protein machine called chaperone. Chaperone is a protein that can fold the primary protein or refold a misfolding protein to get a normal protein, or if the system failed, we can have the protein enzymes to eliminate the damaged protein. So if the system is wonderful, we have a lot of good proteins. If the system is damaged, we have skin problems, skin aging, and accelerating the aging system. So this is the optimal turnover. But we live in a world with oxidative damage, and this ROS can produce protein misfolding. And when we have this damaged protein, we can have aggregates and fibrils. Like we can see in cutaneous amyloidosis, these fibrils can deposit in dermis and causing disease. There's many papers discussing these damaged proteins in skin problems. Psoriasis have a lot of TNF. TNF trigger ROS, and ROS can induce cytokines, inflammation, and protein carbonylation. Of course, we can see this in atopic dermatitis. We see a lot of protein carbonylation in atopic dermatitis, and even in skin cancer. There are research that we can found a lot of carbonylation in the skin when you have melanoma. And of course, in the skin aging, there's a different pathways related to skin aging, but when you see at the bottom of my slide, the loss of proteases is one of the most important part that we can understand in domination of the aging process. Skin is so difficult to treat because we have external factors and internal factors. Air pollution, ozone, chemical agents, UV radiation, metabolism, genetic factors, all involved in production of the ROS. When you have a lot of ROS, we have this oxidative damage, and the oxidative damage can impose this stable alteration in damaged protein. One is these consequences is carbonylation that can involve the union of carbon monoxide with the protein and producing the aggregates linked with the agent and disease. There is a proportional related with the skin process and the skin aging. When you have a lot of protein carbonylation, we can accelerate the skin, reducing the hydration, reducing the skin radiance, degrading collagen and elastin, and increase the inflammation in the skin. So carbonylation and skin problems are related. But we don't have only carbonylation. We have other products like glycation. We can put the union of a glucose, a sugar, with the protein in a stable way, linked with the same problems, aging process and skin diseases. So maybe one solution is target this first step, target the loss of proteases. We have now an option because the growing evidence that this is acting in gene expression, but also we have protecting the whole proteome will help to build and maintain tissues, allowing to slow down the process of aging. So this is a new chapter in the treatment of the skin. We can use not only antioxidants, but we can improve the machine that keep the proteins fold and keep the proteins producing the correct functions. So this is now the future that comes today. Thank you very much.
Thank you very much, Professor Rocha, and we understand that protecting the proteome is a fundamental discovery for skin aging, but maybe also in the future for other age-related diseases. It is no time for questions. Yes, we have still times for questions, so if you have some, please go ahead, and we have microphones.
Thank you for the nice lecture. I'm from Korea. My name is Narashin, and I'm a dermatologist. I'm actually, I wonder, we are familiar with AGE, not the protein carbonylation. I mean, advanced glycation end product for skin aging more than protein carbonylation. So I just wonder if we're using that method, the protein protection, that can help, advanced glycation end product too.
Okay, I answer. Yeah. It's true that AGE and glycation, as also Professor Rocha said, it's a very, it's something we know that we should target. I just like to remind that glycation is linked with sugar content. Okay, so we need to have contact between sugar and the protein. So it's not for everybody. And also, glycation is particularly detrimental to the proteins with a very long shelf life because they need to have long-term chronic exposure to sugar from the blood circulation. Carbonylation is much more universal. It doesn't need to have sugar to be damaging the protein. It targets all protein, not only those who have a long shelf life. So it's a more universal damage. But it's true that when you reduce the attack by free radicals, you reduce the oxidative environment of the cell, and this will help target glycation. So the two of them are slightly overlapping, but we are really convinced that carbonylation is much powerful because it is much universal. It touches all proteins, and carbonylated protein cannot be repaired.
The key difference is that for glycation, adding a glucose, sticking a glucose molecule to the protein, there's an enzyme called PARK7 from Parkinson, or DJ1, which is deglycase. That means the enzyme comes, sees the sugar sticking on the protein, and takes it out, like, bad teeth. Takes it out, and so it's irreparable damage. And when it does not work, then glycation causes eventually Parkinson. That's why the gene is called PARK for Parkinson, because that's the defect in deglycation of proteins, whereas for carbonyl, it's carbon double bond oxygen. There are too many of these groups in natural molecules. This is keto group. You can't repair something that is normally present everywhere. So it is not reparable, and yet it changes radically the charge and therefore the structure and function of the protein, especially interaction of proteins. So small group, just oxygen, is more dangerous to proteins function than is adding a sugar, but basically because adding sugar is reparable, it's a deglycase, and there is no deoxygenase to take the carbonyl, the oxygen out. So that's the main difference, while following a non-reparable damage is more relevant than following a reparable one. Yeah.
Thank you for your answer. And I have another question. We all know that nowadays the microbiome is the superhero in everywhere. So how you guys think about that with your product and process you use to?
Microbiome, you're right, is a new organ, I would say, to be targeted. So we consider that it's, because we believe that skin is an ecosystem, so it interacts with the microbiome. So we are trying to rate the effect of our ingredient also on microbiome to see how these two territories can coexist. Our ingredient doesn't impact the microbiome because the microbiome is also constituted of proteins, so we don't have any detrimental effect. But to know better how we can interact both territories, we need to go on with the studies that we have just begun now.
In microbiome, we have a problem that is the loss of diversity. So in the future, we need something to improve the diversity in the skin and the gut. We tried to do this with probiotics, but we can use probiotics in the skin as a topical drug. So it's not so simple to improve this loss of diversity.
So what we have done so far is to demonstrate that our ingredient is not detrimental to the microbiome, and then we are willing to dig further in this field. Maybe the topic of a next talk next year.
Thank you. Yes. Other questions? Oh, yeah.
Thank you. I'm a dermatologist from Turkey, and I wonder, especially, for example, for actinic keratosis, we know that in several years it can turn to the SCC. And is there, if we start to use such kind of productions to that people, is there a risk for prevention, or is there a treatment for the prevention, or there is a risk for that?
We try to think this to improve the results using this approach, but there is no paper yet to comprove this, okay? But this is approach, correcting this protein damage to improve the results in actinic keratosis, okay?
You also mentioned about that it's different from the telomerase. It's
proteomes. Yes, it's totally
That's
different for gene strategy.
Okay, thank you.
First of all, I want to thank you for sharing such excellent and very, very intriguing knowledge about this new theory. It's really very, very interesting. And I want to change the point of view and to ask you if there is some investigations which show how changes of lifestyle, I mean thinking, eating habits and physical exercises affects our chaperones.
Go on first? Okay, thank you. I go first. So basically, you want to link epigenetics and proteome?
Yes.
So as far as I know, because the science of the proteome is a very new science, so as far as I know, I haven't read anything about a link between lifestyle, epigenetics, and impact on chaperone activity of our endogenous chaperones. Yeah. To my knowledge, I've never read that, but maybe
there's a
you create the
there's a wide variety of different chaperone activities, from very small one to big structures that are like a barrel, to put bad protein in and then massage it with ATP until it takes the most stable form, which is the correct one. So there are there are aggregases, the chaperones which stimulate the aggregation of misfolded protein, making garbage bags of oxidized, useless proteins in order to prevent their toxicity, actually. So these garbage bags, these big amyloids, are not that toxic. They are extracellular, and they cheat the immune system. Immune system thinks that these are foreign proteins and attacks them, and therefore there's inflammation due to this. But actually the killers, I showed quickly, those, the picture of those small circular structures. These are the pores, and they are, you know, like taking the cork out of the bottle, you know, like this. So they make holes in the cell, and that kills cells. So these small oligomers are more toxic to the cells and are probably the most direct cause of all degenerative diseases caused by the loss of cell mass. And we know many of them, of course, right? So there's a whole zoo, the diversity of chaperone activities, which one stimulates aggregation, then there are disaggregases that take out the proteins when the capacity to degrade proteins comes. So we are at the beginning of understanding how functionally diverse and well coordinated are these surveillance systems for proteins, because that's the only thing that matters. No proteins, zero life activity, bad proteins, bad life activity, and that's it. It's acting directly on the action, which is the action of proteins. Right? Okay, so that's why a little bit this focus is shifting from being obsessed by genes, because it's information. You know, we feel it like some, you know, text, philosophy, poetry, whatever we are. You know, we like complicated things, but actually we are, I hope, in the middle of the process to demonstrate on people's skin, you should, that this theory works in its simplicity.
And for sure, what has been demonstrated, if we talk about heat shock proteins, which are endogenous chaperones, and we know the link between stress, heat stress and UV stress, these increase the level of heat shock proteins in the cells because they are defense systems. Okay, so if you increase the stress, you increase the defense level via heat shock proteins. So this is reported since the year 2000, I would say. But the link with the diet and so on, I don't think it has been very much digged in.
Okay.
But we have to remember the link with diet and mTOR pathway. So mTOR pathway is damaged when you have increase of this pathway. So maybe I agree with you, there could be a link between diet, mTOR pathway, and link with and damage with the proteins.
But then we should stop eating, so it's not a solution.
Yeah. We just have time for a last question. Yes, I see one.
Good morning. My name is Anna Domaszewska, and I am a scientist from Poland. And I would like to firstly congratulate for this wonderful morning session. And I've got one question and one comment. And you, Professor Isabelle Benoit, showed us the graph with the axillus extract comparing with different antioxidant. And what I noticed, it's very similar effect of one flavonoid's quercetin. I'm wondering if you were thinking of maybe adding this flavonoid to this agilis extract to enhance the activity of this product. And my second comment and question is that maybe you think about adding your extract to the exosomes, which are very popular here in this Congress, to protect proteins that are containing in these exosomes. And thank you very much for this session.
Thank you. Well, the first comment I have on your question is that you realized that we are opening a new way. So now the page is nearly white, and for us to write with various possibilities to roll out this concept further. We could indeed associate with an antioxidant. It's true that quercetin performs very well in this in vitro test, but we know the difficulty of stabilizing quercetin in a formulation and the problems of bioavailability. So probably if we wanted to synergize the antioxidant activity of our ingredient, it would not be with quercetin, but we do keep in mind to try to enhance further the antioxidant activity with a synergy with another compound. And the second is we have begun the application of our extract on the skin because for us it is simple, okay? We are experts of the skin, so we know how to deliver the active ingredient where we want it to go in the cells here at the level of the dermis. So formulating for skin application for us was easy because we have years of expertise in this domain. Now much remains to be done if we want to address more particularly to target the ingredient maybe inside the cell to a specific target. And then we have a wide array of possibilities that opens to us in terms of vectorization, encapsulation. And this page remains to be written. Also maybe for Peros application, we can see that oxidative damage is the root cause of all age-related disease. The first one that we target today is skin aging, but the potential is much wider. And probably the galenic form would have to evolve also to better fit all the applications.
Thank you. Thank you. It's now time to close this session. Thank you to all of you for your presence and your time today.